the other curves show you the scattering from the lipids, which again are coincident with where the lipid headgroups where the actual toxin is. So again, the theme here is that we can see reflectometry isolated to different components, and then we can overlay them to see where our different components lay in our system. Diffraction measurements using, in this case, deuteration of selective amino acids can position the molecule orientational-wise relative to the lipid bilayer. So you get orientation and sort of extension depth of penetration. And again, ultimately, it would be this work is heading towards trying to understand this toxin again in the presence of reconstituted ion channels. So the other story to tell, I think is the last story, is the story of these pore-forming antimicrobial peptides, phycedins. Basically, they're peptides that are from fish. There's a number of homologs, and they're again, for the interest here, there was two variants, P1 and P3. The sequences are shown here with very small sequence differences. But the inhibitory potential of the P1 and the P3 were actually quite different, with the P1 being more active against the E. coli than the P3. So again, these peptides are seen through electron microscopy to basically disintegrate membranes and over time. And so the question is, though, is how they actually do that and how they actually disrupt the membranes is still an open structural question. So again, what Ella's group did was basically look at these peptides together with a collaborator who first was interested in doing, there was first